The overall goal of this procedure is to record postsynaptic activity at inner hair cell afferent synapses. This is accomplished by first exposing the sensory epithelium of a cochlea recently removed from a decapitated rat. The second step of the procedure is to position the excised apical turn on a modified cover slip and transfer it to the recording chamber. The third step of the procedure is to locate and gain access to an afferent bouton with a recording electrode. The final step of the procedure is to form a tight seal onto and then rupture the afferent fiber membrane, establishing the whole cell configuration of the patch clamp technique. Ultimately, results can be obtained that show postsynaptic currents or potentials from a single ribbon synapse through whole cell patch clamp electrophysiology.
Visual demonstration of this method is critical as the dissection and preparation of cochlear tissue is difficult to learn and the small afferent fibers are hard to recognize and access.
Prior to beginning the dissection, prepare a modified cover slip that will hold the preparation in place during recordings. Using an 8 to 12 mm circular glass cover slip, apply a drop of half-cured Silgard towards the edge of the cover slip. Place the thick end of a fine insect pin on the cover slip. Hold the insect pin tightly against the glass with forceps. Heat the Silgard against a heated coil to set. Prepare 10 to 20 microelectrodes as described in the accompanying text. After fire polishing with a microforge, the microelectrode should have an outer tip diameter of approximately 1 micron. The final thickness of the pipette wall equals about 1/3 of a micrometer. Following anesthetization and decapitation of a 3-week-old rat, begin the dissection by removing the skin, bisecting the head, and removing the brain to expose the temporal bone. Remove both temporal bones. Place each temporal bone in a clean dissecting dish containing standard extracellular solution. The next step is to remove the bone encapsulating the temporal bone so as to expose the cochlea. Hold the temporal bone securely at the base with a pair of forceps. Identify the round and oval window and the cochlea. Orient the cochlea so that the oval window and spiraling side of the cochlea face upwards. Remove excess bone around the cochlea. Use a second pair of fine forceps to chip the bone directly off the cochlea, beginning with the area that is more transparent than the rest of the bone. Here the bone is thinner and easier to remove. Continue removing the bone from around the apical coil. Use microdissecting scissors to cut through the modiolus beneath the apical turn. Then disconnect the apical turn from the lower turns of the cochlea. Take care to protect the apical coil. It should not be pulled or stretched. Finally, remove the apical turn from the cochlea without tearing the tissue too much. Use fine forceps to remove the rest of the bone. The next step is to remove the stria vascularis and the tectorial membrane. Carefully remove the stria vascularis, the shiny strip of tissue situated outside the hair cell region. Make sure to avoid removing the sensory hair cells, which can easily become detached along with the stria vascularis. Next, use fine forceps to detach the tectorial membrane, the shiny, semi-transparent membrane that sits above the sensory hair cells. Now trim excess tissue and bone, and flatten the preparation with the forceps. This is necessary so that the tissue can be placed evenly under a pin. The dissection is now finished. Place the preparation under the pin on the prepared cover slip, taking care to position the pin away from the hair cells. Once the dissection is complete, use forceps to transfer the preparation to the recording chamber while keeping a drop of extracellular fluid on the cover slip that holds the cochlear tissue. Immediately begin perfusion with extracellular solution to ensure better survival of the preparation. Use the microscope's 10x objective or the 40x water immersion DIC objective to locate the preparation. Orient the preparation so that the recording electrodes can approach the inner hair cells at a right angle to their lateral walls. If the visibility of the basal region of the inner hair cells is limited because the preparation is curled over, use a recording electrode to push the outer edge of the preparation down against the glass cover slip, taking care to avoid pressing the inner hair cells themselves. In this preparation, the upper electrode is holding the outer edge of the preparation down. The lower electrode marks where the afferent boutons are located for reference. The NC70 Nuvicon camera displays the 4x magnified image on the monitor. To assess whether the tissue in the inner hair cell region is healthy, find the hair bundles as a point of reference and focus down to find the nuclei. Focus further down towards the base of the inner hair cells with its afferent endings, and back up until reaching the level of the nuclei. On the monitor, now locate the afferent boutons around the base of the inner hair cells. Boutons are spherical or ellipsoidal, approximately 1 micron in diameter, and are light in color with a shiny appearance. You will find them by focusing up and down. The next step is to clear a path to the afferent fibers. Using a recording electrode with a 1 micron tip filled with intracellular solution, apply positive pressure and use the micromanipulator to maneuver the electrode to the preparation. Use this electrode to make an incision in the preparation at the level of the inner hair cell base. Push the electrode between the thin layer of supporting cells and the inner hair cells, using positive pressure to forge an access route to the area at the base of the inner hair cells. Carefully remove the electrode and replace it with a fresh electrode with a 1 micron diameter tip filled with intracellular solution. Using positive pressure to maintain a clean electrode tip, move the recording electrode through the access hole around adjacent supporting cells towards the afferent bouton. Ensure that the electrode tip is directly in front of the afferent bouton. Boutons offer more resistance to movement of the pipette than the supporting cells and the inner hair cell membrane. This difference can be felt as the pipette is moved. Move the pipette up and down and push to make sure that the bouton moves. This indicates that the pipette tip and bouton are in the same plane of focus. With the electrode pressed against the afferent bouton, simultaneously release positive pressure and apply suction to form a gigaohm seal. Apply gentle bursts of suction to rupture the membrane within the electrode to obtain a whole cell recording. If the cell patched is an afferent bouton, small capacitive transients are observed in the whole cell recording mode. This figure shows typical transients recorded from an afferent fiber. Begin recording synaptic activity. If the afferent fiber does not exhibit spontaneous activity, depolarize the hair cell by applying an extracellular solution with a higher potassium concentration. This figure shows typical current voltage relations for afferent fibers on the left and inner hair cells on the right. The current voltage relations were recorded from a holding potential of -84 mV with voltage steps from -124 mV to +36 mV in 10 mV increments. Voltages are shown to the right of some traces. Focusing on the current voltage relations from an afferent fiber, note that the excitatory postsynaptic currents, or EPSCs, are present during the majority of the voltage steps. The EPSCs reverse to positive at about +6 mV. This recording was carried out in the presence of tetrodotoxin to block voltage-gated sodium currents. Without the blocker, fast-activating sodium inward currents would have additionally appeared in the recording and identified the recording as an afferent fiber recording. Note the slowly activating inward current at hyperpolarizing voltages. This current is not present in inner hair cells or supporting cells and provides a good indication that the recording is from an afferent fiber. These recordings show the current voltage relations for an inner hair cell. The arrow points to the fast-activating outward potassium currents at positive potentials. These are followed by delayed rectifier potassium currents. Due to a narrow pipette diameter and high access resistance, the inner hair cell currents shown here are smaller than expected. To obtain accurate recordings of inner hair cell currents, pipettes with lower access resistances should be used. However, the recordings presented here demonstrate that the current voltage relations of inner hair cells and afferent fibers can be clearly distinguished. This figure shows synaptic currents recorded from an afferent fiber. Note the variable size and shape of the EPSCs. The recordings were done at room temperature with tetrodotoxin applied to block the voltage-gated sodium currents. This figure shows simultaneous recordings from an inner hair cell and a contacting afferent bouton. A voltage step was used to depolarize the inner hair cell, and in response, inward calcium currents were recorded from the inner hair cell. Calcium influx results in neurotransmitter release and activated EPSCs in the afferent bouton. Note the synaptic depression appearing during a 50 millisecond inner hair cell depolarization.
Whilst attempting this procedure, it's important to remember to take care dissecting and preparing cochlear tissue, and to remain patient and optimistic when attempting to record from afferent terminals.